I'm Marc Rothenberg, professor of pediatrics at Cincinnati Children's Hospital, and I'm very pleased to be participating in the Food Allergy Fund Summit today. I wanted to first start off by talking to you about our current situation, which is related to COVID. This has been a very awkward, unprecedented time for all of us, and it's really inspired us to think about life so differently. And this disease, this pandemic that we're encountering has certainly affected the food allergy community in ways that we didn't appreciate ever before. And this has created new challenges, but many opportunities. I want to first give you some background on myself and explain that I'm a physician scientist. I have an expertise in immunology, a PhD and an MD from Harvard Medical School. And I have a long-standing interest in understanding the immune system and how it responds to various stimuli, particularly allergens. But now we see the importance of the immune system as we see how vulnerable we are to the viral infection, COVID-19. I'm personally inspired to see the camaraderie that so many people, communities, and nations have had working relentlessly to confront our ever-growing threat from COVID-19. I started medical school training in the 1980s. And at that time, the AIDS epidemic was the single biggest challenge. It was also caused by a virus. And now it was transmitted incidentally from monkeys into humans, similar to the way the current virus is transmitted from an animal. In this case, it's thought to be bats into humans. But unlike the AIDS epidemic in the 1980s, which took over a decade to identify the causal agent, the human immunodeficiency virus, the COVID virus was discovered within only 1 month after the disease was first recognized. And this emphasizes the importance and advances in scientific medical research and the cooperation of international researchers and people around the world to solve the mysteries of COVID-19 so quickly. The scientific accomplishments that we've seen applied to COVID-19 are remarkable, and it really underscores the long-term investment, particularly by our government through the National Institute of Health. The investment in basic research has helped to defeat AIDS, converting it from a chronic disease, from a deadly disease when I first was introduced to it, to a chronic ailment. I myself have now been impressed by the AIDS epidemic that I saw when I was a medical student, when I learned a lot from the dying and brave patients that were dying from this disease. And this is when my own interest in immunology was born, leading to my own personal impactful career, having contributed to describing new diseases, which I'll talk about, the food allergy diseases called eosinophilic GI diseases, and also developing novel therapies for this disease. In the 1990s, we know the importance of the international effort associated with the Human Genome Project, which involves 6 countries that deciphered the genome. And although not directly related to COVID-19, it was the Human Genome Project that facilitated the rapid identification of the SARS coronavirus type 2 that is responsible for this disease. And this underscores the investment in basic research, which I know is so important to the Food Allergy Fund, has on so many practical matters such as COVID-19. Although COVID-19 has made us humbly aware of our vulnerability, I myself am increasingly impressed by the amazing and rapid scientific breakthroughs that are taking place to confront this disease. And this is encouraging for other diseases such as food allergy. We've seen already over 9,000 COVID-19 research articles published, with one new article now coming out every 5 minutes. The FDA has reviewed nearly 500 new drug trials per week, and they're approving many of them. This is a record-breaking efficiency, and this type of activity is marveled and needs to be sustained. Could you imagine if this type of sustenance of activities was sustained and applied to the food allergy problem? This is part of my hope for the future. It's also remarkable that within 63 days of discovering the COVID-19 virus, that the first subject was treated with a dose of a plausible candidate vaccine. And already now there's over 10 different vaccines that are in testing. Despite much scientific progress, we still have a lot more to do in terms of applying science. And I personally was perplexed to see the shortcoming of implementing widespread COVID-19 diagnostic testing, particularly related to the fact that the testing is relying on a test, the PCR test, that kids in high school today are learning about. I'm concerned more than ever about the next global crisis, another threatening virus, a bioterrorism attack, or a nuclear event, each as predictable as the current COVID-19 crisis. While we focus on the mortality and economics of COVID-19 in the Western world, I have concerns about even potentially more devastating effects on COVID-19 on the poor and indigent. And we've seen that even in the New York region, where certain vulnerable populations are more susceptible to the disease, particularly those where social distancing is harder and healthcare is less avoidable. I hope that our current predicament will spark a new set of young minds, just as mine was stimulated to go from the AIDS epidemic to go into immunology research. I hope that these new young minds will become even further stimulated to solve global health problems related to things like COVID and the food allergy epidemic. After all, what's more important than improving life through medical research? COVID-19 has the potential to unite so many people around the world, as vividly illustrated by the progress that we've made already. The Jewish sage Hillel says, if I am not for myself, who am I? But if I am for myself alone, what am I? And as we responded to the 9/11 crisis with the formation of the Homeland Security, which has been very successful in curtailing terrorist activities in the US, I hope that we can also band together globally to solve this current crisis. And I'm greatly optimistic to see all the developments that are taking place. This leads me to my own personal interest in research that related to food allergy, focused on the emerging set of diseases which we now refer to as eosinophilic associated GI diseases, or EGID. The most notable EGID is eosinophilic esophagitis, or EOE. EOE is a chronic food allergen-driven disorder, which is unlike food anaphylaxis in that it is not an acute reaction, but it's a chronic ongoing constellation of GI symptoms. And it's also unlike classic food allergy, which is elicited by only a limited set of foods, typically one, because in EOE and other EGIDs, the body has a breakdown in tolerance to multiple foods, and the common patient is allergic to all the common food types. The disease was only described 20 years ago, but we've learned so much. We now know that the disease is caused by food allergy. It's associated with a very strong adaptive and innate immune system, meaning that the memory arm of the immune system, particularly T cells, are actively responding to food, and they're making high levels of type 2 cytokines, particularly IL-13. We know that the IL-13 interacts with the innate immune system, particularly the epithelial cells that are lining the esophagus. And it's the interaction of the innate and adaptive immune system that leads to the inflammatory response that's triggered by the foods and associated with a massive accumulation of eosinophils. Eosinophils are the cells that you see in the background here on my background image. Eosinophils are normally white blood cells, normally produced at very low levels. In fact, they're one of the least common white blood cell when you look at the peripheral blood. However, the cells dramatically increase in allergic disease. They're a hallmark of allergies, and they can accumulate in the tissue in specific diseases such as asthma. Also, notably, high levels of the cells accumulate selectively in the tissue in patients with EGID. So if you look at a patient's biopsies obtained by endoscopic procedures, and you look at the biopsy under the microscope, the most common cell that you'll see infiltrating the tissue will be the eosinophil. Eosinophils in the esophagus are normally not present, but in the disease you can have hundreds in one single microscopic field of the biopsy. We now understand that the disease is caused by an interaction of genetic susceptibility elements and early life exposures. So it's an interaction between the genes and the environment. Even identical twins do not have 100% concordance in the disease, but we do know that genetics plays an important role. The environment is very critically involved, and we know that also through genetic studies, particularly with dizygotic or fraternal twins, that have a very high concordance rate compared to regular siblings, indicating that the early shared life environment in utero in the first year of life primes the immune system to develop this disease in the context of certain genetic susceptibility elements. We've identified the specific elements in the genome that confer susceptibility, and unlike our original expectation, where eosinophil was thought to be the primary defect, what we found was it was the epithelium that was the defect, because the gene products that were causing susceptibility to the disease were encoding for epithelial cell gene products, particularly a gene called calpain-14, which is an intracellular protease that regulates the barrier function of the esophageal epithelium. And the barrier is very important because part of the immune system is to actually use barrier surfaces to protect the inside of the body from exposures, whether it be microbes or whether it be a food type that you might be allergic to or may not be allergic to. But we need to remember that food itself is a foreign body. The immune system is greatly challenged to respond to each food that you intake and to actually have a normal tolerance reaction to that food. In the case of EOE, the second most commonly associated genetic variant is in a gene called TSLP, which is a critical hormone of the immune system that regulates the difference between sensitization and tolerance. So the two gene products that we've identified, one regulates the barrier, the other regulates the memory arm of the immune system, controlling tolerance or hypersensitivity. So we now are really beginning to understand the key molecular and genetic events that take place that lead to this disease. We do know that the eosinophil is a very potent cytotoxic inflammatory cell, and that the tissue damage is strongly associated with eosinophil accumulation and activation. And it turns out that the #1 gene in the whole body that's overexpressed in the esophagus of EOE patients turns out to be an eosinophil potent chemoattractant and activating factor, a gene called eotaxin 3. This gene is responsible for the recruitment of the eosinophil from the blood to the esophagus, where it gets activated and has a prolonged lifespan and can release its toxic mediators in the milieu of the tissue environment of the esophagus. This then leads to the constellation of symptoms, whether it be difficulty swallowing, abdominal pain, difficulty with growth because of malabsorption in early life, or vomiting, and food impactions, which is food getting stuck in the throat because the esophageal motility doesn't work normally, or the tissue becomes very scarred and fibrotic, and the esophagus, which is normally wide enough to pass food, becomes narrow and the food doesn't go through. So we have spent a substantial amount of effort to try to understand how we can use this scientific knowledge, just like we are doing with the COVID outbreak, to understand how we can rapidly apply this to improve the lives of patients. And we've initiated a number of clinical studies which are giving us great options and success stories for patients. We now know that we can treat eosinophil GI diseases by 3 major different available therapies. One is to identify the foods that are triggering the disease and remove them. This is often the most common approach, particularly ones that parents like in young children, getting close to the heart of the matter by removing the culprit that's driving the disease, which is the food antigens. Number 2, we often take topical glucocorticoids in the formulation that was originally developed for asthma, and instead of having the patient breathe the asthma inhaler in, we have the patient swallow the inhaler. And what that does is it allows the medication to soak the esophagus and to to suppress the inflammatory response, and they can have a very dramatic effect. Now, that is an off-label indication of the medicine. We are pursuing approval of different topical glucocorticoids, and that's a process that takes quite a while, but it's a very close to finalization, and we're optimistic about that soon being approved here in the United States. We also now know that proton pump inhibitors, which were originally designed for neutralization of acid, drugs such as Prevacin, can be useful for patients with eosinophilic esophagitis and even other forms of EGID, so that this should be considered a therapeutic option in patients. Interestingly, despite the fact that they work so well, we know very little about the mechanisms, and this is a very substantial area of research in my laboratory to understand the mechanisms of proton pump inhibitors and how they block the inflammatory cascade, particularly the allergy reaction. And uncovering this, which we're making great progress currently to solve, can actually help us treat not only eosinophil GI diseases, but other allergic diseases associated or not associated with eosinophils. I want to finally come back to some of the most exciting research that I've seen happen to me in the eosinophil field. When I first started working on eosinophils, we needed to understand how these cells were controlled. And it was back then during my PhD that I contributed to the development and understanding of a particular cytokine called IL-5, which is an eosinophil growth factor, showing that it was overproduced in humans with various eosinophilic diseases. And then spending the 90s working on animal models where we could show that this cytokine could be overproduced and cause eosinophils in preclinical systems, and that we can block IL-5 in asthma models, for example, and then attenuate asthma features. This led pharmaceutical companies to develop humanized antibodies against this eosinophil growth factor, IL-5. And it was in early 2000s when we began to get the data coming back, and it was not satisfactory. The initial studies showed no effect on the asthma, even though the drugs were very effective at lowering eosinophil levels. So it was at that time that the pharmaceutical companies took a hiatus. But as an academic scientist, I didn't want to give up, and I had a strong degree of faith and inclination that this pathway was going to be operational. After all, the drugs were very effective at hitting the target. They were very safe, but we needed to figure out which patients would most benefit. So we spent a number of years applying for grants and trying to get money for these types of studies, and finally able to demonstrate, first in open label trials, but eventually in controlled trials, that the proper usage of these drugs that are blocking the eosinophil growth factor can be clinically useful under the right conditions. And that led to a resurgence of interest in these new drugs, which eventually led to the first approval of a new allergy and asthma medication after 12 years. The last new class of asthma medications was approved 12 years earlier, which was anti-IgE, or Xolair. The new class of drugs, which are initially IL-5 antibodies, neutralizing antibodies, and more recently, the IL-5 receptor monoclonal antibody, which not only neutralizes IL-5, but also depletes eosinophils, has been approved by the FDA. These drugs are all now approved for eosinophilic asthma. They're gaining increasing traction for other eosinophil-associated diseases, and there's a couple other approvals for them. And there's now an active investigation of these drugs for eosinophilic GI disease, again with a focus on EoE, but also lower GI diseases such as eosinophil gastritis. And the results are now rolling in, and the data is beginning to look spectacular. We use the right patients, and we have the right metrics for monitoring how the patients are doing, which, by the way, were all developed in conjunction with patient groups. So we work very closely with patient advocacy groups to understand what's important to them, and then working with other key stakeholders in the field, including the FDA. We've come up with protocols that are ones that are likely to succeed because we're following endpoints that are agreed upon and ones that are within our reach based on the mechanism action of the drug. So when I first started doing this work, we didn't know anything about how the eosinophil was regulated. It was during my early work when we identified key regulators, particularly IL-5, the eosinophil growth factor, and about 5 years later, during my postdoctorate training, identified eotaxin, which was the eosinophil activating factor. Subsequently, experimental systems were built. We developed mice that were missing these molecules and subsequently missing eosinophils, and we proved that eosinophils in preclinical systems were involved in things like experimental asthma. But it was really in the 1990s where these were basically proof of concept studies. And it was only until the last century, starting around 2000, when we began to actually use these biological agents to see if we can improve patients with allergic disease. And it took about 15 extra years to finally get to a stage where we're actually now able to completely control, and in this case, eliminate eosinophils from the body. What's amazing is despite all of the research that's been ongoing in animals showing eosinophils are involved in the immune system, and they're probably important for parasitic infections, and there's even emerging data for their role in cancer, we've found that the drugs that deplete eosinophils are quite safe. And it's very reassuring to know that even young people are beginning to get these drugs, and there's not an emergence of a number of side effects. We do see that the eosinophil depletion is very dramatic. It's often universal, although not seen in every patient. And we're trying to understand which diseases now that are associated with eosinophils are actually mediated by the eosinophil. So what we have now created as scientists is a human system, which I call an eosinophil knockout human system. And this is basically using safe drugs to treat patients that started with eosinophils. Many of them have high levels of eosinophils, and then to remove the eosinophils selectively from the body with very safe and effective and fast-acting medications. This has really opened up the field, made it completely, it brought it to a much higher level, one that I would say is very academic. It's stimulating new minds, just like the COVID crisis is stimulating new minds. And it's also emphasizing that the field of allergy and immunology and food allergy is becoming very molecularly oriented. When we first started, we needed to skin test everyone. A lot of things were just diagnosis, but limited treatment. Today, we know that there's a number of now FDA-approved drugs, such as the A-Immune product, such as the biologics that I talked to you about. We also have Xolair, another biologic, and there's a series of other drugs that are on the horizon that are truly going to have an impact. And I see what type of innovation is emerging from my lab and my colleagues' labs and those around the world. And a lot of this does involve critical interactions with the drug companies, many of them both big pharmaceutical companies that are taking an interest, but also very impressive startup companies that have ideas that they're trying to pursue. So it's been my privilege to participate in all forms of research related to food allergy. I have a passion for basic and translational research, and my goal is to learn from each and every patient through research, but to make sure we provide the best care for everybody and to make sure that we learn from everybody at the same time. Great progress is being made. I can tell you stories and stories about people that are very thankful and leading very thriving lives because of the breakthroughs in science that have been applied to them. And I know that the horizon is very bright, and I look forward to working together with you, getting to know the people associated with this organization and the summit, to understand the critical factors that are important for you, to learn from you, because I learned so much by talking to every person, particularly learn new ideas from very smart parents and patients themselves. And I just want to close by saying I'm very optimistic about where medical research is taking us. For those of you young kids, I can really say that by the time they go to college, the landscape for treatment of food allergies is going to be dramatically different. And that's all because of the support that we're receiving through organizations like the Food Allergy Fund. Thank you very much for your attention. My contact information will be available. please do not hesitate to contact me. And if you're interested, I can put you on our mailing lists for information. I wanted to also let you and your colleagues know, not only patients, but colleagues in the medical field, that we do put out all of our data in a public forum. We have a web server that we set up called EGIT Express, and this allows people around the world to get instant access to our data as it's generated and verified, so that we can find out what other ideas are and make sure that the information we're releasing is optimally used by people around the world. Thanks again for your attention, and I hope to get to meet you. Have a nice day.